Now, I have the pleasure of introducing today's keynote speaker, Dr. Amalia Harrington, UMaine's newest lobster biologist. In a very short period of time, Dr. Harrington has made her mark on campus. Following her doctorate and a postdoctoral fellowship here at UMaine, she took on the pivotal role of regional coordinator for the UMaine Sea Grant's American Lobster Initiative, where she was instrumental in establishing the backbone of that critical program. I am so incredibly pleased that she has become a faculty member in our School of Marine Sciences and an affiliated faculty member of our Lobster Institute, although I think that one was pretty mandatory. From her early research on California spiny lobsters to her current position at UMaine, Dr. Harrington has demonstrated a deep commitment to the science of lobsters and its translation to the betterment of coastal communities. We are excited to have her leading this discussion tonight, and her talk is titled, Lobster in a Changing Environment, Breaking Down Barriers Through Collaborative Research. Please join me in welcoming Dr. Harrington.
A little shorter, hold on. Okay. Well, thank you very much for that introduction, Dean Rowland, and thank you all for being here, and also to the co-hosts for inviting me to participate and share some stuff with you tonight. In particular, kicking things off for the symposium, which I'm really excited to see all of the great talks tomorrow. So again, I'm Amalia Harrington. I'm in the School of Marine Sciences. And today, I know it's a little bit late, so we're not going to talk about a lot of data, but I am going to provide sort of a holistic approach to, why is this not working? Where do I point it? Uh-oh. I have a PhD, I swear. Oh, there we go. Now it's working. Sorry. Okay, so I'm going to give you a little bit of background about myself, touching a little bit on my introduction to lobster, and really thinking about collaboration and ecosystem change as a unifying theme for all of us here over the next day or so. And then I'm going to really dive in and highlight a couple of what I think are really good examples of collaborative work that I've been able to be a part of since I started at UMaine, as well as some other upcoming projects that I have in the pipeline to see if you guys are interested in, collaborating, which is great. And then finally, we'll end tonight really planting some seeds for the next day to really have you guys thinking about where we want to go as a research network in the face of change. So, as Dean Rowland mentioned, I started my introduction to lobster in California, looking at California spiny lobster research with Dr. Kevin Hovel. And with my master's, I was really focused on understanding habitat use and sheltering behavior inside and outside of newly implemented marine protected areas. So I had the opportunity to work on this larger initiative, which was really valuable for me as a student because I got to do a lot of great stuff as a master's student. And one of the most formative and exciting pieces of this was a tag recapture program aboard commercial vessels with lobstermen in partnership with Cal Fish and Wildlife managers. So this is a really exciting project where we got to look at movement patterns and population demographics in these MPAs. But really, my larger contribution to this was developing and then executing a dive-based survey program to understand not only thinking about where the lobsters are, how many there are. They aggregate because they like hanging out with friends, unlike American lobster, but that's okay. But also, getting an assessment of the habitat type, so thinking about substrate and the macroalgal community. And with these observational studies, I really got to develop some questions and some hypotheses to then test with some manipulative field and lab experiments. So I had a whole series of work where I did tethering experiments, so think lobsters on leashes, to look at how shelter construction as well as group demographics affects survival of juveniles. So this was all scuba-based in the field, and I followed it up with some mesocosm experiments to look at shelter choice in the presence and absence of predators. So again, I don't want to talk about the data or the results of this. You can check out the paper. I'm happy to chat later on. But I bring this up because this was a really critical stage in my life as a scientist and really gave me the tools and the confidence to continue moving forward in the field of lobster ecology. This was the first time that I participated in research, not only to have a poster or a publication or a paper, but to really contribute to applied science and have impacts down the road in thinking about reserve efficacy. This also was really important because it introduced me to the wider world of lobster ecology. So yes, I read a lot of papers about spiny lobsters, but also slipper lobsters and clawed lobsters, and really got to know a lot of the great research here at the University of Maine to really continue to inspire me in this field. And probably most critically, and what I hope to instill with you all, as well as in my own lab, is it really made me want to work with other people to do team science. So this was a great opportunity to interact with not only scientists from Scripps, which is kind of a big deal on the West Coast, that was great, but also researchers inside and outside of my unit at SDSU. I mentioned the fishermen as well as managers, but this was also part of a much larger effort, the MPA monitoring enterprise, where we had scientists involved in a variety of different species, a variety of different habitat types, all coming together to understand the impacts of these MPAs. So really this experience was really great for me, and I decided I wanted to continue working in this space. But so then I moved to Maine and got my introduction to real lobster for you guys in the room. Threw that in there for you guys from Maine. But so I moved to Maine in 2015 and joined the UMaine community, but I didn't start my dissertation work until 2016 with Dr. Heather Hamlin in the School of Marine Sciences and also in collaboration with the Aquaculture Research Institute here. As Dean Rowland said, I then transitioned into a postdoc position, and then I stayed at UMaine but switched gears and ended up at Maine Sea Grant, where I was involved with the American Lobster Initiative as our extension specialist on that project. And then last year, in 2024, I started my position. So I am, as Dean Rowland said, affiliate faculty with the Lobster Institute, but also part of the Maine Agricultural Forest and Research Station, or MAFES. So I've had a really great opportunity here at UMaine to kind of experience everything that my unit, as well as the university and the wider space in lobster has to offer. And I'm really excited to continue working in this space in a new way. But regardless of which role I've played here at the university, my work has also, like always, centered around understanding how environmental change affects lobster as a model fishery species. So you guys are all familiar with the life stage, hopefully because this is a lobster conference. But what I'm really, really jazzed about is thinking about these transitional stages. So what are the trade-offs when it comes to settlement? What does it cost to be an early benthic juvenile versus an adolescent, adolescence to adulthood? And more recently, really thinking about the trade-offs in a changing environment when it comes to reproductive output. And environmental change is a really broad aspect. We could talk about a whole bunch of different things. But as you know, the Gulf of Maine system is a really great place to work if you want to think about change and ecosystem change. So I lied, there is one data figure here, but it's not mine, so it doesn't count. But so whether you're thinking about the physical environment, so increasing temperatures, changes in carbonate chemistry, or changes in dissolved oxygen, the Gulf of Maine has changed for everybody in the physical environment. But along with this, we also have changes in the biological factors, so thinking about range expanding species and how on top of this physical environment changing, we have all these new species and new interactions to think about. It's just a really excellent place to work, and using the lobster, you can touch on all different aspects of this kind of change. And I'm really excited in looking at the program for all of you guys presenting tomorrow, because I think whether or not you know it, everybody is focused on change in some way, shape, or form. So this is a word cloud with everybody's titles. Again, lobster's in the middle, so you're in the right place. Everybody here is thinking about lobster. But we also can see some things in here that are really indicative of change. So thinking about the future, thinking about temperature changes, simulations, all kinds of aspects of change, no matter what your expertise is. There's something for everybody here, and I'm really, really excited about that. What's also really exciting, if you look at the program, at least to me, is that a lot of this work has either directly or indirectly grown out of the National Sea Grant American Lobster Initiative. And I just want to touch on that in a really brief moment here. For those of you that are not familiar with the ALI, I'd highly encourage you to take a look at the story map collection that I and my colleagues have put together for this initiative, because it does a really nice job of taking this idea of lobster in a changing environment from a scientific perspective and really boiling it down to an understandable communications product. And again, if you look at the different themes that we have under here, there's a lot of change, a lot of topics, all related to understanding how we can increase the resiliency of this important fishery in the face of ecosystem change. So there's a lot of really great connections, a lot of great partnerships that I was able to form while I was at Maine Sea Grant. And just to reiterate kind of the depth and the breadth of this initiative, this is an overwhelming bubble diagram to just really show you all of the different entities, people, different groups that were all involved in this initiative, whether we're looking at the research program or the regional lobster extension program, which included individuals from all of the 7 Northeast Sea Grant programs. It's just a really great group of people all thinking about change. And you can see here that Maine Sea Grant was in the middle of all of this. And so while I was in my position, I had the opportunity to interact with everybody. It was a really fantastic time to think about sort of matchmaking and connecting people working in the same space, but also building off collaboratively from one round to the next to really advance the science. And what's really critical and important to my work now at UMaine, I worked a lot with Chris Cash to think about how we can better integrate scientists at the University of Maine and those that are affiliated or not yet affiliated but potentially interested in being part of the Lobster Institute, bringing them into this wider network. So if you haven't had a chance to talk about or think about the ALI, I'd highly recommend you reach out to Michelle Brown, who is now the Lobster Research and Extension Coordinator at Sea Grant. So there's Michelle, yay! Have a moment to chat with her if you haven't yet, because there's a lot of really great work still happening here, and she would be the best person to give you some insight as to where we're going with this. So as I mentioned, this was a really great opportunity for me as an individual to build out my network and make some partners. And I kind of want to highlight a couple of projects that I've been working on, also funded through the ALI that we started last September, that I think are really, really exciting examples of kind of that iterative building of research over time, but also bringing in new people to understand this resource in a changing environment. And the first project that I want to talk about is one where we're really trying to do a more holistic understanding of characterizing molting hormones to better predict the probability of molting. So this is a really fun project that has a really good group of people. I think it's good because I'm a part of the team, but anyway. So we have, including myself at UMaine, Dr. Heather Hamlin in the School of Marine Sciences. We have an excellent PhD student, Emma Dillart, as well as a fantastic undergraduate intern, Madison Brown, working on this. And our co-PI on this project is Dr. Heather Glahn, who's at Maine DMR. And really exciting, we have a ton of collaborators on this project. And most exciting is Erin Legacki, who's at the USDA. She is a salmon person and has a really great background in vertebrate hormone analysis, so bringing her into the fold has been really exciting. We also have Tracy Pugh, Dr. Tracy Pugh from Mass DMF here. Again, I'm so sorry, Tracy, I could not find a picture of you. But so she's the logo. So we've also had some great support from the Lobster Institute on this project, as well as a couple of contractors working on this at Maine DMR. Evan Barron has been really taking the lead on this, but Declan McCormick has also been involved in this. And then we have a couple of staff at USDA who've been helping with the lab analysis, Ollie Rice and Daniel Boyd. So this is a really great team of folks that I've really enjoyed working with. But really the driver for this is that, we all know that lobsters grow via molting. This is an incremental process that has a lot of factors involved, but we still don't really have a great understanding of growth, particularly in these larger individuals. And what's really great about this project is that it builds off of some a great research study that Tracy did in Massachusetts looking at growth in large offshore lobsters, then the pilot study that Heather did at Maine DMR that we're really building off here in Maine. And it's just a really great opportunity to kind of see what Tracy did, try to expand upon that and add some extra layers here. And if you're interested in thinking about or hearing more about the larger context of this study, I would urge you to see Heather's talk tomorrow at 10:15, where she'll be talking about the larger molting study here that this is a part of. So This is a really exciting project that adds a couple of layers of including some new indicators of molting. But really, just to give you an idea of what we did, if you didn't check out Emma and Madison's poster, it's the pink one that's right back there. But I will walk you through some of our timeline, just in case you didn't get a chance to look at it. But so the first thing is we need lobsters, right? So we worked in collaboration with Ready Seafood and Bold Coast Seafood to purchase adult lobsters, as well as the sea samplers at DMR to get some of the smaller individuals. Once we collected them, we brought them back to DMR's lab in Booth Bay and did a pretty intensive intake of all of the animals. So we measured them for a variety of different morphometrics, took the traditional external characteristics that we think might be good indicators of molting. So thinking about, blue tinted meris, the mottled abdomen, also looking at spine wear and the age of the shell, but also taking a pleopod off of these animals and doing some staging under the microscope. Where this is somewhat new, we took a hemolymph sample here, so a blood sample of our lobsters. We took this at intake as well as an image. Once these guys were deemed worthy of being in our experiment, then we put them into a semi-natural environment off the dock at DMR, where they were held in traps, and we kind of, let them do their thing. Emma and her team and Evan, a lot of the hard work of checking every day to make sure everybody's all right. But really the big intensive part was every Wednesday from when did we start? April? And until now, we're still going. Every Wednesday going out and pulling up all of the traps and sampling all of our animals. So with this, we pull them up. If they haven't molted, we take them out. We do a blood sample, do another hemolymph sample, take some of that to do a Brix measurement to get an idea of roughly the protein content, then take a picture of them, as well as another picture of the pleopods so we can go back and do some staging later on. So these guys, if they didn't molt, we did this, we put them back in the water, let them do their thing. If an animal did molt, we didn't bother them, we let them stay there. But once an animal went through a molting event, we gave them 10 days to kind of recover, harden off their shells before we bothered them. And then we went through and we took basically an outtake panel of all of these indicators at the end. So what we're left with is a whole bunch of photos and hundreds of samples of hemolymph. But this is really where the USDA comes in and where Erin really has her chance to kind of shine and help us figure out how we want to move through this process. So what we're doing with this is a liquid chromatography tandem mass spectrometry, or LCMS-MS, to get an idea of hormone profiles here. So we have all these samples. I bring them up from the coast. I bring them up to Orono because Erin is based here on campus, thankfully. We run them through, after we optimize them and we extract the hormones, we run them through Lightning, which is the LCMS-MS that they have over there. It's a fantastic machine that's really fun. And then it spits out something that looks like this, which is a chromatogram showing you not only the peaks, but also retention times of these different hormones. So Erin and her team, as well as Madison, have done a really great job optimizing our six different panels for our hormones. So we're ready to go as soon as the government opens to really hit the ground running and do all of our samples. So we're really, really excited, and we've done a lot of the groundwork. We're just chomping at the bit, ready to go. So we have this idea as sort of one novel indicator here, and we also have all these pictures of hemolymph color. And Emma and I had a really great conversation with Laura Crane, who with her colleagues published earlier this year a paper where they used green crab hemolymph color as an indicator of molting. So we had this great conversation, and Emma's made some really fast steps to kind of take that protocol and modify it to give us a really great quantification of these colors. And we have a new undergrad who's going to be helping with this. So we're ready to go and add this along with our LCMS-MS data to combine it with these traditional metrics that we think are good indicators of molting, to throw it all into a model and see what we come up with as the best predictors or the best candidates for us to think about molt timing and probability. So our plan, if everything goes smoothly, is to run all of our samples this winter and start building out our model in the new year. That's subject to change, right, as things are still somewhat fluid. But then next summer, we really, even if we don't have the model fully built, we are still going to continue expanding our data set and increase our sample to really get a better swath of sampling to continue refining our model. So with this, again, I didn't show any data. You can check out the poster. But if you are interested in hearing more about this, please reach out to Emma, as she has done a fantastic job coordinating all of these different pieces so that we have a really great data set to then work with. Sorry. I did anyway, I'm sorry. So that's one really fun project that, again, has kind of built off of some of the work that's been funded with the ALI. And I should have said, Tracy's project that I referenced earlier was funded in, I think, 2019 or 2020 iteration of the ALI cohort. So it's just been really kind of fun to continue that process as well. And then the other project that I really want to spend a little bit more time on is one that's being led by Dr. Heather Glahn that's really focused on understanding growth during the first year of lobster life. So this is a fun project where we have Heather leading this in collaboration with Jess Waller, who's also at DMR, who I think most of you know. But our team here at UMaine now includes myself, Dr. Michelle Staudinger, and then Chloe Root, who's a master's student on here, who just took on a capstone student. I'm sorry, I don't remember their name, but also some great undergrads working on this project. And again, we have two really fantastic contractors from DMR working on this. Declan is really leading the charge on this, and Evan has provided some additional support as needed. But also critically, we have Russell Brewer, who's been helping with some of the field sampling, as well as Robert Russell, who is also helping with the sampling, but is also just generally a fantastic person to work with a really positive attitude. So if you ever need to do field sampling, I would highly recommend you work with him because he's just a really nice person, and I enjoy working with him. But so with this project, we kind of have three overarching goals and objectives. The first being to really continue expanding Maine DMR's larval survey. So you can see in this picture, Declan and Chloe are hard at work sampling the larval survey. But then the next thing that's really, really exciting, that again, Chloe has a poster. You should check it out if you haven't had a chance yet. But Heather came across this really cool tagging method that was being used in crayfish. And she said, hey, why don't we try it with our post-larvae? So that's what Chloe and Heather and others have been doing all summer. So you can see that's somebody's finger or thumb, and that is the abdomen of a post-larva with a little tiny P-chip. So it's a really fun thing that Chloe's been doing in the lab to kind of see if this will work out, but also trying to get a better handle on understanding growth and development phenology. So again, reach out to Chloe if you have any questions about the lab portion. Heather is also the best person to talk about with the larval survey. But again, really what I want to touch on with this is another component that a lot of you have been engaged with and a part of so far in the last year. And this is something that builds upon some workshops that we did while I was still at Sea Grant, thinking about largely in the lobster research community, what are the research gaps and the needs and how we can move forward across some of the challenges that we're facing as a group. So with this, instead of thinking holistically across all of the fields of lobster biology and ecology, we're really focused in here on early life history stages. So we did one workshop over Zoom and then another one in Burnt Island, which you can see David took a fantastic photo of us. Everyone's really happy, although it was freezing. So this has been a really fun opportunity to really dive into the challenges, data gaps, but also best practices for working with early life stages. And what's really exciting about this is this has culminated in a really fantastic manuscript to guide research and monitoring of early life stages in lobster. So this is still in the works. I'm going to show you a couple of things that are part of the manuscript. They're not yet published, so please don't take any photos, just in case you're going to want to. I know you will. So I should mention that we have, I think, like 25 co-authors on this, and Heather has done a really great job wrangling everybody to make sure we hit our deadlines. But you can see from our table of contents in here that we have a pretty diverse array of methods and approaches here that we touch on, including a whole section on field, a whole section on the lab, and then what I think is most fun, the biological endpoints section. And we really kind of set this up to be a choose your own adventure. So you open the document and you're like, hey, I have this question. What kind of methods can I use to address it? What are the endpoints I could get? And it's just been a really, really fantastic collaboration with all of these great folks that are active in this space, including individuals from a range of career stages, including some students as well as postdocs. It's just been a really fun opportunity. And what I really want to show, this is the part that you probably want to take a picture of, is highlighting one of the really great aspects of this is that we brought on a graduate student at UMaine, Madeline Young, who is not only a great scientist, but a fantastic artist. And she drew this illustration for the paper that shows all of the different field methods that we use. And it's just a really great rendition of all the great science, but a really great example of how we're trying to think more creatively of blending people's strengths. And Maddie has a really fantastic artistic eye, and kind of lending that to us in this early life history has just been a really fantastic opportunity. So again, we're still in the final stages of this. I think we're in pretty good shape to hit our deadline, or our target deadline of a submission in December. So keep your eyes out for this. It's really fun, and I think you'll all find it a really great resource for you, as well as to share with anybody who's interested in getting into this field. So I also put Maddie up here because she's a good transition into some of the other projects that I'm working on here, just to give you a flavor of the kinds of research that I'm including in my portfolio, to encourage you all to reach out if you're interested, because I am super excited to work with anybody and continue collaborating and building my research network. And Maddie is working on a project right now that is looking at understanding the impacts of bait and diet in general on lobster thermal physiology. This has been a really fantastic opportunity for me because Dr. Gail Schwieterman, who's in the School of Marine Sciences, was one of the first faculty in SMS to really reach out and say, hey, let's collaborate, let's work on something together. She's a fish physiologist, so fish have backbones, which I have no idea what to do with, but kind of blending her expertise with my interest in lobster. Maddie's done a really good job of kind of being the glue that pulls us all together with this project. And what's also really great about this is with this project, I was able to take on my first undergrad capstone, Alyssa Benoit, got it right? Alyssa Benoit, so they graduated in the spring of this year, did a fantastic capstone project, adding some capacity to Maddie's project, but then stayed on this summer as a research technician at the Darling Center to continue working with Maddie and really adding another layer of support, which has been really great. So it's been nice to kind of see her transition from, Maddie's taking a real ownership over the summertime, and then Alyssa is really just helping to continue making sure that we make this progress work. And again, I'm not showing data, but check out Maddie's talk tomorrow at 1:20 because she has some really interesting results, especially in light of some of the posters that are over here. So I think it's going to be a really great session. Oops. The other sort of space that I'm starting to dip my toe into is thinking about emerging contaminants. We have a PhD student who finished in May, Crystal Boley. We're in the process of getting her results and her dissertation out, looking at imidacloprid as a contaminant with lobster. But what I really want to highlight here is something that's really getting a lot of attention here at the University of Maine, and that's thinking about PFAS. And this is particularly relevant to today's conversation because I was only able to do this work through a direct communication from Chris Cash, putting me in touch with Dr. Charles Rolski, who's at the Shaw Institute. He is the executive director there. He's been working in the PFAS space for a really long time and really was eager to try and get into the space of marine organisms and invertebrates. And so he and I have been pretty successful in getting a couple of seed grants through the university, namely a Maine SMART grant through Maine EPSCoR, as well as some seed money through the UMaine ARS Center of Excellence, which is a really great initiative here on campus, all focused on emerging contaminants with a particular focus on PFAS. So with this, we did some pilot work, gathering some baseline information on larvae as well as adults in the natural environment. And what's really exciting is that we were able to do some pilot exposure experiments over the summer to look at how short-term exposure affects the behavior of post-larval lobsters. And I brought in Dr. Bobby Moorfield, who's a postdoc at the Aquaculture Research Institute, who has some really great expertise looking at behavior in sea lice. So we kind of took his methods and protocols and tweaked them to work with lobster. We also brought in my colleague Robert Harrington, also at ARI, who helped not only build a bunch of our systems to help make sure we could hold our animals, but he's also an analytical chemist and really helps me when I can't do the simple math to think about concentrations. So it's been really nice working with this team again, building out, bringing in people that otherwise wouldn't be in the lobster space and kind of bringing them with me and helping them and leaning on them for their expertise. The final project that I want to talk about is something that is really, really new, and we just put in a proposal last week to try and get this funded, and that's with Dr. Megastapa, who's also an SMS, but she's an oceanographer. And this really came up as a car ride. We were driving from Orono to the Darling Center, and we had just had our last workshop with the early life history group, and we were kind of just chatting about things that we still don't know about lobster, and it came up, we really don't know a lot about diet in these early benthic juveniles. So Meg suggested, well, I do sediment flux. We could think about, delivery gravitationally from the surface to the benthos and really get a better handle on diet in early life stages. So we ran with that. We put in a proposal. We're hoping to get this funded, again, with support from the Lobster Institute. So a lot of really fun areas of research that I hope you reach out to me if you're interested in. But I also just want to mention one sort of infrastructure update here on campus that I am extremely excited about. So if any of you had a chance to kind of walk around campus or drive at all, there's a lot of construction going on. But the one area that is really, really exciting and worth notice is a construction of ARI's Sustainable Aquaculture Workforce Innovation Center, or SAWIC. So it doesn't look like this yet, but it's really close. We've got, some frames and part of a roof, which is really, really exciting. So when I got my position here in the School of Marine Sciences, one of the first things I did was talk to Dr. Debbie Bouchard, who's the director of ARI, to think about how I can invest some of my startup funds to build out an environmental stress lab at this facility. So lobsters are not aquaculture species, but that's okay. We can still do some recirculating aquaculture to do multi-stressor experiments. So I've been working with Robert Harrington to rebuild a multi-stressor lab that we used to have here on campus that got demoed where this new building's gonna be. So I'm really excited. We're thinking about building out a system that can be somewhat modular, so thinking about multiple life stages of lobster, but also other species, and most importantly, thinking about training students, graduate and undergraduate students, and thinking about partnerships moving forward. So a lot of really great stuff on the line and in the works here. And again, I'm just really, really excited for you all to be here and really want you to continue thinking, as you spend all day tomorrow listening to these really great talks, thinking about collaboration and ecosystem change, where do you think we should be going as a lobster research community? And I ask this because we've been having a lot of these conversations since I started at Sea Grant and now in my new position. And regardless of who I'm talking to, when we think about where we need to go, what we want to do as a network, networking really comes up in making sure we have the right partners. So making sure that we have those key partners to help foster new connections and new collaborations, whether that's from the resource perspective, industry, or management, really thinking critically about how you want your science to be used to help increase resiliency for the fishery is going to dictate which kinds of partners you need to have on your team. And now more than ever, given our current landscape with funding as well as the status of our country, collaboration is really going to be the only way that we can move forward. There's a lot of crazy stuff going on, right? But that doesn't mean we can't do the science that we need, the climate science that we need to do. Maybe we just rephrase how we do it. But we also need to start thinking more creatively about making the best use about the funds that we do have access to. So really thinking strongly about working with partners to reduce duplicative effort and really critically thinking about how we can support the students that we need to continue doing this research. So in particular, making sure that we don't lose out on the next generation of scientists. So thinking more holistically about how we can maybe piece together some funding for students across multiple projects not only allows us to have students, but also increases their exposure to a variety of different skill sets and tools, making them better prepared to enter the workforce post-graduation. So if you look around this room, I stole this map from Chris Bream on the website for the event. We have a lot of people here represented. So we do have folks from four provinces in Canada as well as 7 states, and we have a huge diversity of expertise in the room. So I know there was a lot of conversation during the poster session, and I hope that this continues, but really try to reach out to somebody new. If you're afraid of introducing yourself to somebody new, I'm happy to do that. I don't know everybody, but I'm happy to pretend that I do. So please try to reach out to somebody new, and over the next day, really start to think about how you can do, if you're not already doing this, because I know a lot of people already are, think about doing those cross-institute collaborations, and also really critically, and what's great about this is thinking about doing some cross-border collaborations. And if you are really excited about working with your friends, either to the north or the south, I would highly recommend that you join us in January of next year for the town meeting. This is going to be in Moncton, and it's a really great opportunity to hear issues that are relevant to both Canadian and American fishermen. And we have a really great list of panels set up and some really great speakers that we hope to come share their knowledge with us all. If you are not affiliate faculty, but you are interested in that, I would highly encourage you to think about joining. Again, as Dean Rowland said, we have internal affiliate faculty, but also external groups, and Chris has done a really great job of building our connections there. She's also been doing a lot of great work bringing in graduate students. So if you are a graduate student and not yet connected with the Lobster Institute, please reach out to Chris and get on our email list and really give us some feedback on what you would like to see us do from the Lobster Institute perspective to really make sure that you are getting what you need to be prepared to enter the workforce. So with that, I know I shared a lot of information, but hopefully it's not too heavy because it's not a lot of data, although that one slide, I'm so sorry. But I do want to acknowledge all of the people in California that really helped shape the person that I am today. I would not be here where I am without the team that really helped me with my master's work. And this list of collaborators at Maine only includes the folks that I am currently actively working with, so there's a lot more people, there's a lot more funding sources that have supported me throughout my journey here at the University of Maine. And in particular, I included the Sea Grant ALI network. There are a lot of you. I couldn't list everybody, but I tried to grab as many logos as I could. And I am truly thankful for all of the opportunities I've had here at the University of Maine. And I'm just really excited to hear more from the work that you guys are doing tomorrow and to continue thinking about how we can work together to advance our field. So thank you very much for your time, and I hope you're not too tired. So thank you.